Okay, so I've titled the talk, I should point out first actually that the work in this talk that I'm presenting is actually from Tona Majekwu. He's a doctoral student. He's funded by the West African Agricultural Productivity Program, which is a World Bank program. Okay, so we're calling it the unseen invasion, and there's two reasons for that. The first is that although Nile tilapia has been invading South African rivers for several decades, we actually don't know the state of invasion. We don't know where they are. We don't have a systematic national overview of which systems have been invaded by Nile tilapia, which is a very disturbing state, and it's causing a lot of friction between fish farmers and, for example, the Department of the Environment. Over the fish farmers want to know where they can put their fish farms. The second reason is that it's essentially a genetic invasion. It's an invasion by hybridization. So a lot of the invasion might be genes passing through populations without necessarily being easily detectable, but nonetheless it results in the displacement of indigenous biodiversity, and the genes that are coming into the system from Nile tilapia, into Mozambique tilapia for the most part, would be genes from widespread farmed fish. So it's a genetically bred forms of fish, and displacing the genes in the native species, which is Mozambique tilapia. Okay. So here's the local, Mozambique tilapia. I should apologise, I've left my voice somewhere on the high felt a couple of days ago, so you'll have to bear with me. Okay, so Mozambique tilapia is indigenous to coastal eastern South Africa. This is a predictive model of the distribution. It doesn't match exactly to where it is, but it's pretty good overall. And characteristics of so South Africa is a critical country for conservation of diversity in Mozambique tilapia. It's been listed on the IUCN Red List as near threatened because of hybridization with Nile tilapia, but it has a number of characteristics. Particularly, it's tolerant of low water quality, it's cold tolerant, it's tolerant of highly saline conditions. It can even breed essentially in seawater, actually in water that's more saline than seawater. It can disperse through the sea between estuaries, which are characteristics which Nile tilapia does not have. All of those characteristics are lacking in Nile tilapia. I should also mention that there's 5 other indigenous species of Oreochromis in southern Africa, and we're also concerned with them. But those species tend to be closely related to Mozambique tilapia, and they tend to be restricted to particular regions. So I'm going to talk mainly about Mozambique tilapia because it's the widespread species. Okay, and the invader, Nile tilapia. It's native to the Nile system, to the Niger system, the Senegal, so North Africa, parts of East Africa, parts of Central Africa. It's not native to Southern Africa at all. It's an aggressive competitor, and it grows rapidly and it matures rapidly. The males in particular mature at a much smaller size than Mozambique tilapia. And what we think typically happens is that when a Mozambique tilapia is breeding, the males of Mozambique tilapia are basically crowded out by large numbers of much smaller Nile tilapia, and so there tends to be this bias in the breeding towards offspring that are fathered by Nile tilapia, at least initially. There's high juvenile survival. It does readily acclimatise to new environments, although there are limits. And there's evidence from other places of Nile tilapia displacing indigenous species. There is also actually evidence in other places of other species of Oreochromis displacing native Nile tilapia as well. In general, Oreochromis do hybridize readily when they're brought into contact with each other. And there is hybridization between indigenous species in southern Africa as well. So there were some early introductions into southern Africa of Nile tilapia. Most of the introductions are associated with fish farming, or a sort of interest in fish farming. So someone brings a few fish in and throws it in a dam, and it's in a system initially. But it became more serious in the 1990s when they were discovered in the Upper Limpopo, actually in a key hole farm that's in Mapungubwe National Park. And then later in the Lower Limpopo, this is in Mozambique, near the junction with the Olifants River, where there are fish farms, and hybrids are being detected in the Limpopo. So where does the pressure come from? It comes from fish farming, as I've said about 3 times already. So there's a long history of farming Nile tilapia. It was farmed by the Egyptians. It was introduced outside of South Africa to Southeast Asia, I think by the Portuguese, maybe also the Spanish, but it was introduced over 300 years ago into places like the Philippines and Indonesia. That accelerated in the 20th century, and essentially it's introduced into systems worldwide. This is a map from the FAO. It's a bit old, of the major producer countries. I don't know why South Africa is on this map, because South Africa is not really a major producer country. There's about 5 million. Currently, there's 5 million tons of production per year. Almost half of that comes from China. South Africa produces a few tens of thousands of tons at the moment. And if you coloured in the countries that are the consumers in here, then basically you can paint the rest of the world. It's global. It's not only Nile tilapia that's introduced globally. Mozambique tilapia has been introduced globally, and also another species, Orochromis aureus, has also been introduced. The blue tilapia has been introduced globally as well. So they're widespread. In 1997, there was a genetically improved farm tilapia developed in the Netherlands in conjunction with several other institutes in Europe. And this strain of Nile tilapia has twice the growth rate. And Nile tilapia is already starting from a higher growth rate than most other indigenous or most other species of Orochromis. And so this is the only viable fish for farming economically. It has efficient production. That 0.77 is, you put in for every kilogram of protein that you put into a fish farm, you get 77% back as fish. That's phenomenal. Nothing's going to match that. This is going to feed the world. At the moment, over 50% of the fish that we eat globally is farmed fish. Tilapia is the second most common freshwater farmed fish. So there's a couple of salmon and a couple of marine fish which are above it, but it's one of the really big industries, and it's been growing exceptionally rapidly in the last few years. There's been a bit of a pause in the last couple of years because of a disease that's gone through some of the stocks in China, and also because of declining prices. But the reason that it is expanding so rapidly is that it's because of this efficient production, because the costs of setting up a facility to grow the fish are low, and the costs of running the facility are also low. I don't know if you'd call it tasty. It's like hake. It's actually it's a fish that doesn't have a fishy taste, and so consumers like it because of that, and the meat looks good. So it looks like a marine fish. So it's something that's saleable. And 3.5% of all fish consumed on Earth is Nile tilapia, essentially. So, and the industry is worth about $4 billion a year, of which a pittance is going on in South Africa, and people see that there's potential here. That's why people want to farm it. That's why they want to make farms all over the country, and that's why it's being spread around. Okay, so what's the role of genetics in this? How do I, myself and Tonna as geneticists, see us contributing to understanding what's going on in the conservation of Mozambique tilapia in particular? Well, the first thing we can do is just simply describe the pattern, describe the natural resource that we've got, natural genetic resource in Orochromis. How much diversity is there? What's the distribution of that diversity? From that, we can start to infer why is that diversity, and particularly importantly, we can see whether there are cryptic species. That is, are we underestimating the number of species that we're dealing with here? Secondly, what we would like to do is to find out, well, because it's an unseen problem, because it's a problem of genes being transferred between species, we'd like to know how extensive is that problem. When populations are hybridized, how hybridized are they? How extensive is the genetic replacement with the Nile tilapia genes, and also the geographic extent of invasion? So we'd like to know what's happening to the genetic integrity of Oreochromis mozambicus, particularly if you think that in a lot of, all rivers end up almost all rivers end up at the sea. A lot of rivers in South Africa are quite cold in the winter, and so these are conditions where Nile tilapia can't survive. Mozambique tilapia can survive, but what do hybrids do? Especially hybrids that have backcrossed several times to Mozambique tilapia. That may potentially open an avenue for those genes from Nile tilapia to spread more widely than Nile tilapia themselves could spread. But on top of that, there is also the fact that Oreochromis have been moved around. Other species of Oreochromis, including Oreochromis mozambicus, have been translocated around the country. So there is some loss of the original genetic integrity in terms of genetic structure, and there has been some hybridization between other species of Oreochromis that naturally occur in southern Africa, and there may have been natural historical hybridization among these species. So we need to know the background there. Why do we want to know that? Because we want by understanding the process, we'll be able to inform policy. In particular, we'll be able to say where are the best places for reserves where you should not be able to farm, where are places where they're so invaded that it's not going to change anything if there are fish farms set up. Okay, there's been a couple of phylogeographic style studies previously using a variety of markers which have shown hybridization and informed us actually to some extent about what's going on. Ideally, what we're looking for when we do these studies is we'd like comparability, in that we'd like, if I do a study here and you do a study over there, that we're counting in the same way, that we can compare the data that you got. We'd like replicability so that we can redo it and get the same results, or at least we've got equivalent results. And we'd like a baseline reference. Okay, I'm going to have to move on here. So the problem is that some of these markers, these are microsatellites, a commonly used marker. The problem is that there's low comparability across studies and there's low replicability across labs. That is, you can compare metadata, you can compare the results of the studies, but I can't take your raw data, combine it with more raw data, and get a more inclusive result. And the other problem is there is no baseline reference. By this I mean, what was the background that we should be comparing it with? What's the state that how do we interpret these results? Okay, and there's been a number of studies that have talked about technical hurdles. I might just jump through this so I can get through the result. But the point is this: because tilapia, because Oreochromis is so widely spread around the world, most of the studies that have gone on have been outside South Africa. They've been outside the natural range, and so they're talking. So the individual fish that they're talking about are potentially hybrids already. So when a study from Hawaii compares wild-caught Nile tilapia with Mozambique tilapia found in Hawaii, how does anyone know what Mozambique tilapia in Hawaii should look like? Okay, so what did we do? We took wild-caught samples from rivers all over southern Africa and a few other locations just to pick up some taxa and the reference baseline. These are samples from South African Institute for Aquatic Biodiversity in Grahamstown. We sampled all the species of Oreochromis that naturally occur in southern Africa. We included some hybrid samples, and as a reference, we used 4 aquaculture stocks of Nile tilapia and of Mozambique tilapia. Okay. These are the results from mitochondrial barcoding. So what we have here is a whole lot of samples that have been analysed. These are DNA sequences. have been clumped together. The depth of these triangles shows you the diversity within a group. The little circle here shows you the support. Basically, a black circle means it's extremely high support. And the distance between there and the next black circle is how differentiated this is from the ancestor or from the next sort of group. What you can see is actually the results fall out very neatly. This is unexpected because previous studies have said you can't use this technique for identifying lineages of Oreochromis. The reason for that is simply because they're from overseas. So every time someone picks a fish, it looks different, doesn't match the phenotype at all, but that's because they're probably hybrids already that they're looking at. So we come up with seven strongly supported lineages, and what's of most interest here is that fish that are currently regarded as Oreochromis mozambicus fall into two lineages. So we've got this lineage here, which is very shallow, and then we've got this lineage here. I haven't put in a map of where these lineages actually occur, but this lineage here is basically Limpopo, Kavango, Zambezi, Kunene. It's basically central and west. This lineage is coastal from the Eastern Cape right up through KZN, through the entirety of Mozambique. There is some overlap in the lower Zambezi, in the lower Limpopo, but there's minimal overlap actually in the distribution of these two lineages. So the fish that are being called Mozambicus in KZN is this group. The fish that are being called Mozambicus in Limpopo are largely this group. Another thing you can see here is that all the other species in southern Africa, all the other endemic species to southern Africa, are basically indistinguishable from Oreochromis mozambicus that you get in the Limpopo River on this technique. They're morphologically distinct. Okay, I'll skip through this. What we know from incorporating our data, which was the previous slide, with all the data, all the available baseline reference data, is our tree falls into this bit, basically. So there's one lineage that is basically a diverse lineage which is Mozambicus related. There's a lineage which is Niloticus related, and there's a lineage that's Oreochromis aureus related within Oreochromis overall. I won't say anything more about that. I'm just going to jump through to the conclusions. Okay. So, in terms of our objectives, in terms of what we want to do, yes, there's 7 distinct, well-supported lineages. Mozambicus, there's 2 extremely divergent lineages that we're calling Mozambicus, which barely overlap in distribution. There's been a proposal that the rivers going into the wild coast of the Eastern Cape should be the fundamental reserve for Oreochromis mozambicus overall. Well, that's not a viable idea. That would only conserve one of these 2 lineages. So one entire lineage would be left for invasion to go ahead. We did not detect a single hybrid in the whole study. And the reason we didn't detect a hybrid is because we were looking at a maternally inherited marker. You get mitochondrial DNA from your mother. All standard barcoding is based on mitochondrial DNA. And the method simply isn't capable of identifying hybrids, particularly if the father of the hybrid is the invader rather than the mother. And so this method, which is actually being touted as the method for detecting hybridization in Nile tilapia, fails completely. It's not possible to use it for that. And there's also other hybrid situations which you just can't pick up, which occurs. So what's needed? Well, we need to look at other genes. We need to look across the genome. We need to look at a whole lot of genes. We need to characterize hybrids in terms of what part of their ancestry is from Niloticus, what part of its ancestry is from Mozambicus. And by doing that, we'll identify the natural genetic resource, and we'll be able to identify which areas are suitable for farming, which areas are suitable for reserves. Thank you.